In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to JAX Tech Talk with the technical information scientists of the JAX Laboratory.
Welcome to episode number 66 of our JAX Tech Talk, Let's Talk Swiss Outbred Mouse Diversity. JAX Tech Talk is a monthly 15 to 20 minute podcast style webinar where we explore topics that our customers frequently ask about. For anyone watching live, the chat is disabled, but please submit your questions through the Q&A button. I am Kat Chappelle, and with me today is Katie Zuyazin. We are technical information scientists at the JAX Laboratory. Our role is to serve the research community by providing research education and offering technical solutions and answers to all of your inquiries. Some of you may recognize us from contacting our group or from attending one of our webinars. In 2021, we introduced you to our colony of Swiss Outbred JARC mice. Recently, researchers at the JAX Laboratory were part of a study quantifying the diversity found in our JARC and JADO populations. Originally, Dr. Kelly Brackett was going to join us for this JTT. However, she was unable to attend today. Dr. Andy Sheely, the JAX Laboratory's senior scientific advisor and one of the authors for this study, has graciously agreed to join us in discussing JARC diversity, this study, and what this means for research with this strain. Thank you so much for agreeing to join us today, Andy. It is always a pleasure to speak with you.
Thank you. Glad to be here.
In our first JARC JTT, we touched on the background of our JARC mice and Swiss outbred lineages. Could you remind our viewers about the origin of the Swiss Webster strain and our JARC?
Oh, yes, happily. So what we've prepared for you here is a family tree that kind of illustrates the interrelatedness of a variety of different outbred and even some inbred strains of mice. So JARC mice share their ancestry with a variety of outbred stocks, and this includes Swiss Webster, CD1, CF1, and NMRI. Their ancestry can be traced back to 2 males and 7 female Swiss mice that were imported to the Rockefeller Institute in 1926. And so this family tree gives you a sense for just how many different outbred stocks and even inbred strains that descend from this relatively small number of mice that were imported in the 20s. So the ARC stock was established at the Animal Resources Center in Australia in 1991, reinfused with new breeding stock in 2005, and finally imported to the JAX Laboratory in 2020. So at that time, we received 64 male and 64 female mice, which were used to create 32 different breeding lines or breeding pairs that we rederived, cryopreserved, and used to establish our JARC outbred colony.
So Andy, you touched on some of the other Swiss outbred strains that are available in the mouse world, such as that CD1. How does our JARC actually fit into that lineage with the other well-known Swiss outbred strains?
Yeah, so they are part of a lineage which is shown in blue on our family tree here that passed through the Fox Chase Cancer Center, and the lineage is now commonly known as ICR or CD1. So they will be more closely related to CD1 or ICR outbred stocks of mice, but since many generations of breeding and many years have elapsed, they're likely to be genetically distinct from those other stocks, but they still share many of the common research applications.
So what makes our JARC strain stand out from other available Swiss outbred strains?
Yeah, so initially we received random bred mice from ARC that we converted into a breeding system which is called the Poily method. And so this is a way of rotating breeders through 32 maternal lines. And so this system helps to maintain genetic diversity that's present in the population while meeting customer demand for the model and minimizing the coefficient of inbreeding. So our JARC colony is also maintained at our highest health status and what we call a maximum barrier. And so this is a room that excludes all tested pathogens and opportunistic agents. In addition, and what we will spend a lot of time discussing today, it is the subject of a peer-reviewed publication that describes the genetic diversity that's present in the JARC stock, in comparison with another outbred stock we have at the Jackson Laboratory, the diversity outbred or JADO population. So with ARC, we now have a multi-purpose, JARC, we now have a multi-purpose outbred stock of mice that's maintained with the same quality standards that we apply to other strains at the Jackson Laboratory. And so now this enables researchers to source their common inbred, outbred, and specialty mutant strains from a single source.
So Andy, you did just mention your paper, and we definitely want to kind of discuss that. So you are a co-author on one of the recently published studies that looked at the heterogeneity in that JARC strain. You did use an interesting strategy to do this. Could you talk about that a bit with our viewers?
Yeah, I'd be happy to do so. So this study was conducted to evaluate the allelic diversity in this new outbred colony. And we wanted to use that information to define a set of genetic variants that we could use as part of a future genetic monitoring program, right? And so how we went about doing this is through exome sequencing of 32 males and 32 females following the rederivation and establishment of this colony at the Jackson Laboratory. So it was at the third generation of breeding. And so we wanted to evaluate all of the protein coding sequences in the genome. We use these results to estimate inbreeding, to determine the level of heterozygosity, and also get a sense for how much variation there is among individual mice in our outbred JARC colony. So it was really important for us to molecularly define this strain because there's actually surprisingly little known about the genomes of outbred stocks. So this approach is a more comprehensive way of measuring allelic diversity and estimating heterozygosity compared to other genotyping approaches, which are used at other facilities and seen described in the literature, which tend to focus on a much smaller set of genetic markers.
Great. And then in figure 2 of this paper, there's a good illustration of the diversity in the founding members of this colony. Can you talk to us through that figure and tell us what it means for the diversity and use of the strain in research?
Yes, happily. So this slide is describing heterozygosity as one measure of genetic variation in a population of mice. So a genetically diverse stock is expected to have many more heterozygous genotypes throughout their entire genome compared to an inbred strain, which is assumed to have essentially no heterozygosity in an inbred strain. So this slide is showing individual heterozygosity in each of the 32 male and 32 female JARC mice that we performed exome sequencing on, in comparison to 10 males and 10 females from another outbred stock at JAX, the JADO or diversity outbred stock, which was purposely bred to combine and maximize the genetic diversity from 8 distantly related inbred strains. So it has a completely different breeding history from any of the outbred Swiss mice, including JARC. So we expect JADO to be more genetically diverse on account of its breeding history. So to compare the heterozygosity between JARC and JADO mice, we focused on genetic variants, unique genetic variants that were common between JARC and JADO, these two outbred stocks. Specifically for this slide, we were focusing on genetic variants like single nucleotide polymorphisms or SNPs and small insertions or deletions or indels in the coding sequence. So there were about 120,000 of these genetic variants that weren't present in the C57 Black 6J reference genome. So for each variant, we then determined the number of mice that had heterozygous genotypes, okay? And so in the left panel, you'll see that on average, with each one of these bars representing one mouse, the heterozygosity in one mouse that was analyzed in the study, you'll see that on average, 25% of those genetic variants were heterozygous in individual JARC mice, compared to around 40% in the more genetically diverse JADO strain or diversity outbred strain. So the right graph is a histogram plot that's showing the same data shown on the left figure, right? And so you'll see that the histograms are shifted to the right in the samples from the JADO mice, which reflects the increasing amount of heterozygosity at these different SNPs and indels we evaluated in the coding sequences. But you'll also get a sense that there's a greater variation among individuals in the JADO population, which is kind of what we expect. So overall, we were very happy to see that there is a good amount of outbred characteristic retained in the JARC colony, less so than we saw in the JDO colony or stock, but that was to be expected. So we were happy to find that, there was little to no evidence of inadvertent inbreeding throughout the development and maintenance of the JARC colony.
You always do such a great job of explaining information, Andy. Thank you. So as you mentioned, the JDO does have quite a bit more diversity than our JARC. So why might someone choose to use the JARC over the JDO model?
Yeah, that's a great question. So at a population level, you'll see that JDO has a much larger number of genetic variants and greater allelic diversity, right? And this is because of the very deliberate way in which this stock of outbred mice was produced to maximize genetic diversity from many different, or 8 different inbred strains, right? And so that makes the outbred JDO mice particularly useful for mapping the genetics of quantitative traits. But the development and maintenance of our JDO colony is much more complicated. It involves many more breeding lines and the use of computer software to minimize inbreeding among those lines. So it's not just random mating, it actually takes it a step further. So as a result, because of this complex way of maintaining the JDO strain, those mice are only available periodically. But because we use a different breeding scheme to produce JARC mice that still preserves heterogeneity in the JARC strain, those mice are actually available continually. So not just in waves like the JDO mice. So if we focus on the heterozygosity plot that I discussed a moment ago, you'll see that you get maybe half the diversity at that marker, but only at about 1/3 of the price of the mouse if you compare JDO versus JARC, right? So the lower level of heterogeneity compared to JDO mice means that fewer JARC mice are necessary in a study group to capture the full range of segregating variation we see in that population. We also see less variation in JARC in certain phenotypes or traits, including body weight. So there's less variation in the JARC stock. And so overall, we were very happy to find out that the JARC stock is indeed outbred. It's going to have less genetic variation than the JDO, but they're ideal for these research applications that call for a robust and diverse population of mice. And so this would include safety and toxicity testing, as well as other applications like use as sentinels in an animal health monitoring program.
Thank you, Andy. Many of our customers choose to maintain colonies of our strains in their own facilities. And maintaining the diversity seen to the level that we see in JARC model and most outbred models with a smaller population can be very tricky. Do you have any advice that they can give to the customers on what they should and should not do?
Yeah, so if you've ever taken an evolution or a population genetics class, you'll know that smaller populations are more likely to lose their alleles over time due to random chance and the process of genetic drift. So you need a larger population of animals, particularly for an outbred stock, to maintain the genetic diversity that's present in there. It's also challenging because these are essentially closed colonies, right? And so these are always at risk of losing genetic diversity over time, just simply due to random chance. This happens inadvertently. And if you order or receive a small number of animals to breed in your facility, that sets up a population bottleneck, and breeding in-house can accelerate this loss of genetic diversity. So to illustrate this with an example, according to one model from population genetics, we are expected to lose about 50% of our genetic diversity in our population within the next 50 generations, simply due to random chance and fixation of alleles to homozygosity. So we have strategies in place to minimize the effects of this, right? And this includes the cryopreservation and recovery of founder lines and other approaches. But if someone were to order, say, a small number of mice, like 10 males and 10 females, and try to set up a breeding population in their own laboratory, it's expected that they would lose about 50% of their genetic diversity in much less time, in as few as 15 generations.
Thank you, Andy. And this is a lot of great information on JAX Swiss outbred mice.
Absolutely.
And thank you for coming today for the JAX talk. It looks like we might be out of time. And so we're gonna finish up. If you're curious to learn more about genetically diverse mice, we have a mini course called Mouse Genetic Diversity, Translating Discoveries to Humans. And the link to the course will be added to the on-demand version of this webinar. And if you still have questions, please feel free to contact us at mice.jax.org. And this will conclude our JAX Tech Talk. Thank you, Andy and Kat, and all you listeners for spending this time with us today.
Thank you, everyone. Have a great day.
Bye.